Hi, welcome back to Biochem Talks. In this video, we discuss about Krebs cycle. Krebs cycle provides energy to cells through aerobic respiration. Krebs cycle was first identified by Hans Adolf Krebs. The first and final product of Krebs cycle was citric acid, which is tricarboxylic acid, in short TCA. So it was also named as citric acid cycle or TCA cycle. In eukaryotes, Krebs cycle takes place in mitochondria. In prokaryotes, Krebs cycle takes place in cytoplasm. Krebs cycle is the final common pathway for the oxidation of carbohydrates, lipid, and protein because glucose, fatty acid, and amino acid were metabolized to acetyl-CoA or intermediates of Krebs cycle. Glucose is converted to 2 moles of pyruvate by glycolysis in cytoplasm of cells. Pyruvate enters into mitochondria through proton importer, that is, pyruvate is transported along with proton. In the mitochondria, pyruvate is decarboxylated to acetyl-CoA by pyruvate dehydrogenase complex enzyme. That is, carbon dioxide is removed in this reaction. This reaction is irreversible, that is, forward reaction will later take place. This is because this reaction is exergonic. One molecule of NADH is formed in this reaction. Once the acetyl-CoA is formed in mitochondria, it enters into Krebs cycle. Acetyl-CoA reacts with oxaloacetate and forms citrate by citrate synthase enzyme. This reaction gives water and reduced coenzyme as byproduct. This is the first irreversible reaction of Krebs cycle. Citrate is isomerized to isocitrate by aconitase enzyme. This reaction takes place in two steps. Citrate is first dehydrated to cis-aconitate, that is, water is removed, and again rehydrated to isocitrate, that is, water is again added. This reaction is reversible, that is, reaction takes place both in the forward and backward direction. Isocitrate is first dehydrogenated to form oxalosuccinate by isocitrate dehydrogenase enzyme, that is, hydrogen is removed. Krebs cycle gives one molecule of NADH through this reaction. Oxalosuccinate remain bound to enzyme. In enzyme, it is decarboxylated to form alpha ketoglutarate. This is the second irreversible reaction of Krebs cycle. Alpha ketoglutarate is converted to succinyl-CoA by oxidative decarboxylation using the enzyme alpha ketoglutarate dehydrogenase complex enzyme. Krebs cycle gives NADH second time through this reaction. This is the third irreversible reaction of Krebs cycle. Succinyl-CoA is converted to succinate by succinyl-CoA synthase. During this reaction, one molecule of GTP was formed. GTP was formed by substrate, so this reaction is called substrate level phosphorylation. Krebs cycle gives one molecule of GTP through this reaction. This reaction is reversible. Succinate is dehydrogenated to form fumarate by succinate dehydrogenase enzyme. Krebs cycle yields one molecule of FADH2 through this reaction. This reaction is reversible. Fumarate is converted to malate by the addition of water across the double bond by fumarase enzyme. This reaction is reversible. Malate is dehydrogenated to form oxaloacetate by malate dehydrogenase. Krebs cycle yields third molecule of NADH through this reaction. This reaction is reversible. Let us see amount of ATP is produced when one molecule of glucose is completely oxidized. Through glycolysis, glucose produces 80 ATP. For detail, please see glycolysis video which I have given earlier. When pyruvate is converted to acetyl-CoA, one molecule of NADH was formed. Since two molecules of pyruvate were formed from one molecule of glucose, two NADH were formed. One NADH gives 3 ATPs through electron transport chain, so this reaction gives 6 ATPs. Krebs cycle yields 6 NADH, 2 FADH2 and 1 GTP. NADH gives 3 ATPs through electron transfer chain. So 6 NADH gives 18 ATPs. FADH2 gives 2 ATPs through electron transfer chain. So 2 FADH2 gives 4 ATPs. Through substrate level phosphorylation, 2 GTPs were formed. 2 GTPs is equal to 2 ATP. So total 38 ATPs are formed from one molecule of glucose when completely oxidized to carbon dioxide and water. Thank you. If you like, please subscribe to this channel.